Right. So from fragments to function, a journey through computational metagenomics workflow. So we start with a grand biological puzzle. A single environment sample like soil or gut swab contains the DNA of thousands of microbial species. Shotgun sequencing shatters these genomes into billions of short, anonymous fragments or reads. So the fundamental challenge is how to identify the species and understand their functions from this massive jumbled data set that we have. So our roadmap, a step-by-step journey from data to insight. So we'll follow the data through a structured computational pipeline. Each stage would build upon the last and progressively turn the raw fragments into actionable biological knowledge. So we have basically have raw reads here. We have raw reads, then it goes to assembly pipeline, then goes to binning, then for strain analysis, and finally for biological analysis. Right. So moving on, first we piece together the jigsaw puzzle. Why? The challenge is to reconstruct original genomes from millions of overlapping short reads, a task that is complicated by sequencing errors, repetitive regions, and the presence of multiple closely related species. So we need to introduce two main strategies. That is first, reference-based assembly, mapping reads to a known genome, useful for variants of known species but biased towards the reference, and also de novo assembly, that is assembling reads without a reference, essential for discovering novel organisms, which is our primary focus. Now, the dominant approach for de novo assembly relies on building sequence graph from reads, specifically using the De Bruijn graph method. So the logic of assembly from k-mers to De Bruijn graph. First step is k-merization, that is the assembler breaks every read into short overlapping words of a fixed length k-mers. So basically these are k-mers, right? Here k is equal to 4. Step 2 is to construct the graph. So a De Bruijn graph is built. Here each unique k-1 sequence becomes a node and the k-mer itself becomes an edge connecting the two nodes. So you can see here that we have the De Bruijn graph. Each unique k-1 sequence becomes a node, right? Each sequence that we have chosen here has become a node, and each of the k-mer itself has become the edge connecting those two nodes, right? The assembler traverses this graph to find unambiguous paths which represent continuous sequences of the original genome called context. Practical challenges in assembly, trade-offs and tangles. So the k-mer size dilemma: longer k-mers are better at resolving repeats but more susceptible to sequencing errors and less sensitive for low abundance species. Shorter k-mers are more robust to errors but create a more tangled graph with shorter contexts. Right. Now, some common issues: bubbles are caused by small variants, SNPs. So this is called a bubble. Tips are basically short dead-end paths often caused by sequencing errors. So a tip would be representing an alternative ending or sequencing error, and a bubble represents a small strain variant. So the problem is that assemblers often remove these structures, but they can represent true rare biological variation. Right. So now we need to sort the pieces of the strains that we have achieved by genome. So we need to take the use of binning. So binning relies on the principle that contexts from the same genome share intrinsic properties. So the two most powerful signals are sequence composition, that is genomes have unique compositional signatures like GC content and tetranucleotide frequencies TNF, and abundance covariation where contexts from the same genome should have similar abundance read coverage across multiple samples. Now this is binning in action, the MetaBat workflow. So MetaBat is actually a very useful tool that effectively integrates TNF, tetranucleotide frequencies, and abundance information to bin context, especially across multiple samples. So first we take samples from multiple sites or times. Then the context from the co-assembly are basically arranged in this format. Then we calculate the tetranucleotide frequency TNF, and we calculate the abundance as well. So we get the graphs, and then we get the distance which we store in our matrix. Finally, we get the iterative clustering and final bins, right? So that is the function of MetaBat. Now, quality control: are these genomes complete and clean? So the output of binning are metagenome assembled genomes, MAGs, metagenome assembled bins, sorry, genomes. So their quality can vary greatly. Then we, for basically checking the quality, we use CheckM. That is a standard tool for assessing the metagenome assembled genomes quality using single copy marker genes, that are SCGs, which are expected to be present exactly once in a complete genome. So we calculate two key metrics. First is completeness. What percentage of expected marker genes did we find? High is good. And contamination: how many SCGs, marker genes, that is single copy marker genes, did we find in multiple copies, which suggests contigs from different species were incorrectly binned together. So low contamination is good and a high completeness is better. So this is the MyMac quality standards. MAC quality, basically two parameters or two key metrics: completeness and contamination. For high quality, we have greater than 90% completeness and contamination less than 5%. Medium for this is greater than equal to 5% completeness and less than 10% contamination. And this is basically a threshold that we have set, right? So that's it. Now moving on, part 3 is strain analysis. So looking deeper, not all strains are created equal. So basically, we need to profile strain variation using two main approaches. First is SNP based, where we identify single nucleotide polymorphisms SNPs in core genes to build phylogenetic trees, for example, strain plan. And then we have to check for gene presence or absence. Then we compare accessory gene content across a species pangenome to find functional differences, for example, using PanPlan. So methods for strain profiling, phylogeny versus function. So we first have strain plan, that is SNP based, and PanPlan, which is gene based. So strain plan maps metagenomic reads to a database of species specific marker genes, identifies SNPs, and builds a strain level phylogenetic tree. And the output is it reveals evolutionary relationships and tracks transmission of specific strains. PanFlan, on the other hand, is gene based. That is, it maps reads against a species pangenome and identifies which genes are present or absent by looking for a uniform coverage plateau. The output it generates is basically a presence or absence matrix of genes directly revealing functional potential. And this is how it looks. Now, mapping the ecosystem, who interacts with whom. So, microbes do not live in isolation. They form complex communities and interact through competition, mutualism, and predation. So, if we infer these relationships by building intra-microbiome association networks. In these networks, nodes are representing the microbial taxa, and edges are representing a significant statistical association. So, identifying hub species nodes with high centrality can pinpoint keystone taxa critical for community stability. Now, the basically the payoff is solving the puzzle leads to biological discovery. So we track the disease outbreaks, understand the diet's impact, and improve therapeutics. Right. And to predict growth rates, we can infer how fast the species are growing by analyzing the read coverage patterns across their genomes. So the peak to trough ratio (PTR) compares the coverage at the origin versus the terminus or the end of replication, which provides a snapshot of community dynamics. Right. And linking microbes to the host, so we can find statistically robust associations between microbial composition and host traits using methods like Permanova and WGCNA, right? And this is basically a complete picture where we are basically assembling the genomes, binning them, right? Resolving the strain level differences by performing strain analysis and mapping their interactions to basically get fundamental insights into the role of microbes in health and the environment.